Good morning. My name's Hank Greeley. I'm director of the Stanford Center for Law and the Biosciences, and I would like to welcome you to our conference on prenatal genetic testing. I think we have a really exciting few hours ahead of us because we have a really exciting few years ahead of us. The world is changing in important ways that may ultimately really honestly change our species. So I'm especially happy to do this program here at Stanford today. 2 years ago this month, in May of 2010, we did an earlier conference on non-invasive prenatal testing, looking at a technology that we thought was going to be introduced soon into the United States and talking about issues that we needed to be concerned about. 2 years later, the technology has been introduced into the United States. There are at least 3 companies which are providing these services clinically. One of them, the first into the market, Sequenom, entered the market in October of last year. They estimate that in 2012, they are going to do 40,000 of these tests. And as of the end of the first quarter, their weekly rate was such that on a 52-week basis, they would end up doing 45,000 of them. Now, 45,000 tests may not sound a lot in the context of about 5 million American pregnancies a year, but the best estimates are, or at least my best estimates are, that there are only somewhere between 60 and 80,000 amniocentesis and chorionic villus sampling procedures done every year in the United States. In less than 6 months, Sequenom is already doing as many, almost, roughly the same number of procedures per year as those well-established invasive procedures. And I think that's some evidence that this non-invasive technology is going to prove extremely attractive. Like all new technologies, it will have its pluses and its minuses, and the balance of those pluses and minuses is what we hope to begin exploring today. A few administrative details. Those of you who are looking for wireless access, we have a very intricate and involved, difficult wireless password and username, law school. Just to remind you that you're at the Stanford Law School. I also want to note that the Center for Law and Biosciences is not putting this conference on by itself. We are collaborating with the Stanford Center for Integration of Research in Genetics and Ethics, part of the Stanford Center for Biomedical Ethics, and with the Stanford Journal of Law, Science, and Policy. My hope for this conference is we will have lots of discussion. We have lots of speakers. I'm afraid I am a pack as much as you can and 10% more into any conference organizer. But not only have I asked the speakers to stay, to keep their comments short, but I'll be sitting in the front row with my iPhone used as a timer to try to make sure that we leave plenty of time for discussion, because I think it is the discussion both among the panelists and between the panelists and the audience that should prove to be the most interesting thing of the day. At this point, I'd like to call up the first of those speakers. Oh, one last administrative detail, or not administrative detail. You are at Stanford. This is being put on by Stanford. Stanford has some conflicts of interest here. Some Stanford-owned technology is licensed to some of these companies. I am not even sure which companies are getting which technology, and the way the patent suits and the likely ultimate cross-licensings are going to go, it could be that Stanford will end up on every side of all of the issues. All I can tell you is that nobody from Stanford, from our Office of Technology Licensing or anywhere else, who has any financial interest in this, has had anything to do with the organization of this conference. So, let me now bring up our first speaker. You have bios on almost all of the speakers in your materials. As a result, and in an effort to maximize the amount of time for discussion and minimize the amount of time taken up with things you can otherwise read, I'm only going to provide the shortest of introductions for each of the speakers. I'd like to bring Steve Quake up. Steve is my Stanford colleague in the Department of Bioengineering, and I will just say that he is one of the most frighteningly creative people I have ever met. Every time I meet Steve, he seems to have some new harebrained scheme, which usually then turns out to be successful. So Steve, tell us about the science of non-invasive prenatal testing.
Well, thanks, Hank, and let me just say that you frightened me too. So, So let me talk a little bit about some of the science behind the topic of the day. And it sort of starts with this very simple counting problem, which is to say, as human beings, we're supposed to be diploid and have 2 copies of every non-sex chromosome. But sometimes things go awry and you end up with the wrong number. And this is a karyotype of an individual. You can see chromosomes sort of numbered, and down there the arrow points at number 21, where there's actually 3 copies instead of 2. That's also known as Down syndrome, and it's sort of the most common genetic disorder of live births. And more generally, having the wrong number of chromosomes is the most common genetic disorder, and among those, Down syndrome is the most. And so it affects close to 1% of live births, and the sort of technical term for that is aneuploidy, having the wrong number. So looks like it's pretty easy to figure out who's got Down syndrome and who doesn't, or other aneuploidies. The challenge, however, is actually getting those chromosomes and that genetic material, and in particular doing it before the baby's born. And so that's typically been done through invasive methods, either through amniocentesis or chorionic villus sampling, but both of them involve sticking a big needle right into mom's belly, right next to the fetus, and trying to grab a few cells and recover them and analyze them. And that, of course, poses some risk both for the mom and for the baby to be. And when I became a parent and saw this being done to my wife, both these things being done actually for each of our kids, I began thinking, well, is there sort of a way to ask these questions non-invasively without incurring risk? And it turns out there is. And the basic principle was discovered back in 1948 by these 2 Frenchmen, Mandel and Matthijs. And they discovered that there is a certain amount of DNA that floats around in your bloodstream that's sort of the detritus of dead cells. Normally we think about DNA as sort of the blueprint for the cell, and it occupies this privileged position in the nucleus, which is a little organelle within the cell, so it's in a membrane and a membrane. But it turns out when cells die, they spill their guts into the blood, and the genome gets thrown out there, and it gets chewed up into little pieces. And so there's these little pieces of DNA in each of our bloodstreams that just circulates around. So these guys sort of stumbled across this phenomenon, and this is their data. And it turns out that one of their subjects was pregnant. And they saw in the case of pregnancy, they saw higher values of DNA and RNA. And so that was, sort of the beginning of the whole thing. And this paper was largely forgotten and sort of rediscovered. This principle was rediscovered in 1997 by Dennis Lowe and Jim Wainscoat, who were able to use molecular methods to show that in the case of women who were carrying male babies, there was some Y chromosome DNA floating around in their blood. That's the male sex chromosome. If you're a woman, you shouldn't have that in your blood. And so they sort of reconcluded that, oh, this cell-free DNA some of it must come from the baby. And that set off a sort of decade-long race to try to find ways to turn that into a practical diagnostic. So we eventually cracked that problem here at Stanford, and by we, I mean my former student, Christina Fan, and our clinical collaborator, Yair Blumenfeld, who helped us organize the clinical study. And what we worked out was sort of a universal way to detect aneuploidy non-invasively. So if you take a little bit of mom's blood from her arm, that's very low risk, and it's considered non-invasive in this game. And the principle is pretty simple. It just has to do with counting molecules. And so if you think about the DNA in the mom's blood, most of the DNA is from the mom. A little bit is from the baby, maybe a few percent of it, depending on the stage of pregnancy. And so if you think about a mom with a baby with Down syndrome, that mom will have a little bit of extra chromosome 21 in her blood relative to any other chromosome. Because the mom is putting in 2 copies of each chromosome, the baby is putting in 3 copies of chromosome 21 for every 2 copies of every other chromosome. There should be a very slight imbalance, just a few percent more molecules of chromosome 21 fragments. So if you're able to count molecules, you can measure that difference very precisely using sort of a very basic statistical principle that says your error goes like 1 over the square root of things you're counting. And so you can measure differences as small as you like simply by counting high enough. And so we explored a bunch of different ways of counting molecules, and one of the ones that worked quite well was to use next generation sequencers. And what we did here was take blood from pregnant women. Christina purified the DNA. She then sequenced it on a next generation sequencer, getting about 5 million molecules sequenced, so that's a pretty high number to count to. Each of those sequences was then mapped back to the reference human genome, and it was allowed to vote for a chromosome. And essentially this amounts to looking for voter fraud or overrepresentation of a chromosome. Speaking of fraud. Now I have less authority. So, and this data here shows from chromosome 21 what the results were. The red data points were all from the moms whose babies they were carrying had Down syndrome, and the blue data points are from the moms whose babies didn't have Down syndrome, and you can see that they just totally separate. You can draw a line between them, and so there's just a clear overrepresentation of chromosome 21. Now, of course, there's nothing special about chromosome 21 in all this. You actually, because you're sequencing, randomly get data from all the chromosomes, and so sure enough, you can go and look for aneuploidy of any other chromosome. And in our study, we had 2 subjects whose babies had trisomy 18, and you can see that those 2 are outliers there, and one whose baby had trisomy 13, that's an outlier there. And so you could just sort of read off any of the most common aneuploidy, or indeed any aneuploidy. So we published this in 2008, as Hank mentioned, and it sort of set off just a huge amount of interest in the field. And in the last year, several large-scale clinical trials published, some academic, some from companies, and thousands of women have now been tested with this approach, and it's, as you mentioned, available in the clinic. And so at this point, I should maybe talk about my own financial conflict of interest in being the inventor of this method. At Stanford, patented the work from my lab and then licensed it to 2 companies, Artemis and Fluidigm, both of whom are now called Veronata and Fluidigm, both of whom I consult for. This is probably the first true genomic diagnostic that makes use of the full power of next generation sequencing. And so I was very, very proud of this work. And as Hank mentioned, we organized this meeting a couple years ago, and I went to chat with him. He organized the meeting, and I went to chat with him in his office just before or after. And he's just kind of a real pain in the ass because not satisfied with this. And he says, well, what's it going to take to get the whole genome done? And I said, oh, come on. So went and sort of thought about it, and there was kind of a very brute force way to do it, where if you waited 5 or 10 years and the cost of sequencing got down enough, it would maybe be sensible. But then, we thought a bit more about it. Christina, who had been working on, my student, who had been working sort of on a different project to do what's called haplotyping, realized that there is a way to put together the work she's doing with haplotyping with the work she's been doing with prenatal diagnostics and sort of measure the whole inherited genome in a very sort of slick and easy way. And so let me describe how that works. So the idea is haplotype is just kind of a fancy way of differentiating the DNA you got from your mom from your dad, okay? So you've inherited, one set of chromosomes from your mom, one from your dad, and haplotype is kind of a fancy way to think about that. And so for this baby, it's got sort of 2 sets of genetic data it could have inherited from the dad and 2 from the mom, and those are combined in some unique way. And so, there's, if you think about it very roughly, 4 possible combinations of any haplotype block to work out. And so being able to measure haplotype ends up being really useful because the information here gets transmitted in large blocks, and it greatly reduces the amount of sequencing you have to do to figure out what the baby has inherited. And so here is a way to think about it. The SNPs, which is sort of loosely speaking the mutations you've inherited, are used to define each haplotype block, and they're sort of linked together. And so if you've measured haplotype, you only need to pick out a couple of SNPs to figure out a very large piece of DNA that's been inherited from either the mom or the dad. And it turns out that you can use the exact same counting principle that we used for the chromosomes to count on haplotype blocks instead. And just give me a minute, this thing will, okay. So if you think about what's going on in the mom's blood, for the paternal haplotypes, you just have to look for which one is there. So if one of them is, the dark blue one is represented and the light blue one is not there, well then you can work out the dark blue one is inherited by the baby. So that's pretty straightforward. when you're thinking about what comes from the mom, it's a little more complicated because there's all this maternal background. And so one way to think about it is to say, well, let's just look to see which one is more overrepresented in the same ways that we looked at which chromosome was overrepresented. So if the dark red one is more highly represented than the light red one, you can do the same kind of counting principle and work out that's the dark one that's inherited by the fetus. And so if you have the knowledge of the haplotypes of the mom and the dad, you can, with very little sequencing, work out essentially the whole inherited fetal genome. And if you're a little clever about it, you can actually do away with dad's haplotypes. And if you just use mom's data, you can do almost as well. So Christina went ahead and did some experiments to show that in principle this would work. And she took DNA from these family cell lines. So you have DNA from a mom, a dad, and a child. And she mixed together the maternal and the child's DNA in an appropriate proportion that would be something like what you find in the blood, and was able to work out which of the maternal haplotypes were inherited. And so these are the paternal ones here. So she could work out, which blocks came, so which of the paternal haplotypes, and she color-coded them blue. And same thing for the mom. And so by drawing blocks around which SNPs were well represented from which haplotype block, she could work out what came from the mom. And that is sort of in principle how it all works. Well, I don't know what happened to my slide here. But that is something we sort of published all the details of in Nature Proceedings about 1.5 years ago. Here we go. And so I was showing you initially data from one chromosome. You can do it for all the chromosomes in the genome of the child and work out exactly what was inherited, both from the mom and from the dad and the mom. And so that sort of, in principle, shows how you could do the whole genome non-invasively, which answers Hank's question. Not just doing it non-invasively, but also doing it in sort of a practical and affordable way. And honestly, I think that's probably, in many ways, less interesting than the aneuploidy measurement because there's not as much to act on, although people will undoubtedly probably try to do stupid things with such a measurement. But in fact, the most useful things I can imagine doing with it all involve measurements that aren't linked to any kind of termination decisions or things like that, that are things you'd want to measure in the third trimester about the baby, so you could treat the baby immediately after it's born. Things like metabolic disorders, immune disorders, and so forth that you just would like to know ahead of time because it's linked to treatment of the just born baby. So time will tell, and I'll stop here and leave you with this quote from Maurice Wilkins in case you're tempted in getting involved in this game. Thank you.
We have time for some questions. I should warn you that we are recording this and we plan to put it up on our website at the Center for Law and Biosciences. So anything you say may and can be held against you any place in any country in this world and in any star system in this galaxy, the Lesser and the Greater Magellanic Clouds. If you're still willing to speak, please come up to one of the, I guess we just have one microphone here in the center, to ask a question of Steve. Feel bad making people in the front row move backwards to ask their questions.
Steve, that was a great introduction. You showed that you could haplotype with 17%, which is later in the pregnancy. How early can you haplotype with what percent fetal DNA, and how does that relate to gestation?
Yeah, well, I mean, arbitrarily small amounts, because it's all based on this counting principle. And so it's just a question of counting high enough to resolve the differences. And so it'll work with a couple of percent, with less. It's just a trade-off between how deeply you want to sequence and how well you want to know your result. Yeah.
What were the two companies you mentioned that used the first technique you described? What were the two companies you said?
Oh,
were-
that Stanford has licensed the work to?
Yes.
Yeah, Veronata Health and Fluidigm.
Fluidigm is the second? Okay. Also, I was wondering if there's a measured difference in the accuracy of the identification of the duplicated chromosomes based on the chromosome number. On the slide you showed briefly, chromosome number 22, which I'm most interested in, looked like it didn't have as dramatic a split between the red and the blue data points.
So for the haplotyping approach, is that the question? Or the aneuploidy approach? Yeah. So because you're randomly sequencing DNA, and the chromosomes are different sizes, you, for any given sequencing depth, will sample the larger chromosomes better than the smaller ones. And so 22 is a fairly small one because they're numbered by size, more or less. And so, you know, in relative comparison, it tends to be a little noisier, but you can, again, make its noise level whatever you want simply by going deep enough in the sequencing.
Hi. Did you ever consider microchimerism from previous pregnancies as a complicating factor?
Yeah.
And how can you determine it?
So, you know, we haven't gone out and looked for it explicitly, but, you know, I've kind of done the calculations because people have a sense for what the level of microchimerism is, and, you know that turns out that the majority of the cell-free DNA, a large fraction of it is hematopoietic in origin, meaning it comes from the blood, and so you can work out what fraction of, let's say you're looking at Y chromosome in a male pregnancy, and there was a previous male pregnancy that chimerized. It's a vanishingly small amount. I mean, it's so far below the level of the fetal DNA, which, you know, early in pregnancy, end of the first trimester, it's around 3% fetal DNA, and anything due to microchimerism is orders of magnitude below that. You have to work really hard to find it. Yeah.
This might be a difficult question to answer, but do you have any idea of how long it took to map the fetal genome and how much money it costs?
Yeah, so since I did it, I can say. You know, it took us, well, it depends what point you start counting, all right? I mean, if you start from the point that we worked out how to measure haplotypes in adult humans, from that point to doing the results I showed, you know, it was, you know, probably 6 months of work and a few tens of thousands of dollars. Now that we know how to do it, it's faster and cheaper.
So were you counting your people's time as having any value? I remember when you
No.
sequenced yourself.
No.
Postdocs and grad students are worth something.
I put it into time. You can convert time to money. 6 months of work by 2 people.
Okay. So, Steve, how easy or hard is it in these methods to find a single gene mutation? So let's say you're looking for Tay-Sachs.
Yeah, so, you know, that's pretty simple, and people had done that, you know, versions of that even sort of before our work on aneuploidy measurement. And so you can use all kinds of differential PCR tricks to look for specific SNPs and gene mutations that might be carrier-related things.
What about chromosomal translocations or?
Yeah, translocations are a little more challenging, and I think there's probably ways to do it, but, you know, they don't occur at such frequencies that necessarily justify that.
Multiple repeats?
Yep, so we can...
Just looking for what this can't do,
Yeah.
what this may have difficulty doing.
You know, we've been able to see deletions and things like that, so sort of structural variation one can measure with this approach, and the resolution, again, sort of depends on sequencing depth. You know, one of the more challenging things to work out is twins, and so that's an area where the companies that are doing commercial things, I think, are being pretty careful to try to exclude them by other measurements.
Well, thank you, Steve.